Good afternoon, everyone. Welcome back to the My Tutor Online School. So great to see lots of you joining us already, and hopefully there'll be more of you kind of joining before we get started. Just to say that today is the first time that we're running a GCSE higher biology session, so I'm really excited to get started with that. And a few little bits from me before we crack on, as always. This session will be recorded, so if there's anything that you've missed or anything that you want to go over, then you can have the chance to do that afterwards. And the best way to find the recordings is to make sure that you've signed up to the My Tutor Online School, so you can get emails with links out to all of the recordings. I've seen a lot of questions already about maths today, and I just want to say sorry to everyone who tuned in for maths earlier this afternoon. We had some technical issues with the link, which meant that you weren't able to access that session. So if you did try and access it, apologies again. The best thing to do is we will be running that session again this week on Friday. So make sure you keep an eye out for the timetable schedule on Friday, and you'll be able to join that session again, so you won't miss out on anything at all. Don't you worry. Cool. So back to biology. Today's session is going to be on monoclonal antibodies, and it is going to be led by a tutor called Olivia, who's one of our really experienced tutors on My Tutor, and she studies biomedical sciences at the University of Birmingham. I think she's actually just come to the end of her studies, but she's done lots and lots of tutoring with My Tutor and is a biology expert. So she's going to be leading the session. If you do have any questions throughout the session, then just send them through using the chat function or using the Q&A function, and I'll be able to pick them up for you, or Olivia will pick them up when she sees them as well. So I'm just going to hand over to Olivia now to lead the session.
Okay, hi everyone, thank you all for coming. It's nice to see you all here. So yeah, as we already said, today is going to be about monoclonal antibodies. Now, this is one of the topics that we recommend that you go over really well and that you get a really good strong foundation of before you go on to A-level, if any of you are going on to A-level as well, because it's one of the topics that is tested in higher GCSE biology. And actually the level that you need to know this in at GCSE in comparison to A-level is, to be honest, pretty much the same. So what you're doing now, if you are going on to A-level, is going to be really productive. Now, it's going to be split into two parts. So the first part, we're just going to talk a bit about antibodies, what monoclonal antibodies are and how we produce them. And you can ask any questions while I'm talking through that, and then I'll answer them after that part. And then we'll go on to the second part about the ways in which monoclonal antibodies can be used. And we'll talk about a few of the applications of monoclonal antibodies. So there's lots of tests and treatments which they can be used in, endless applications, to be honest. But you don't need to kind of know the details of any of them. You just need to be able to apply your knowledge. So we'll have a look at some exam questions as well, so you can see the kind of things that you'd get in an exam. Okay. So firstly then, just a quick recap on antibodies generally and the important things that you need to know about antibodies in order to understand this really. So if any of you have been to the other sessions, you should have heard about this, and hopefully you know about antibodies anyway. So antibodies are a protein molecule and they're part of the immune system. So they're produced by white blood cells, and they're produced specifically by a type of white blood cell called a lymphocyte. And that's a key word which you need to try and remember. And there's going to be lots of keywords in this, but maybe try and write them down, write your definitions of them so you can always come back to them. Now, lymphocytes produce these antibodies in response to a particular antigen. Now, what's really important that you understand is antigens generally, when you think about them in terms of the immune system, we tend to associate antigens with pathogens. So they're a part of a pathogen which is recognised as foreign by the immune system, and they're able to trigger an immune response. But actually antigens can be all sorts of things, really. It's just anything that can be detected as foreign by the body. So when we talk about monoclonal antibodies, some of the antigens that we will consider will not just be things associated with pathogens. It could also be hormones, it could also be particular drugs, and anything like that, really. So you can see in this diagram here, this is what our antibody looks like. They're a Y shape, and we've got this part at the top, which our antigen is going to bind to. Now, this follows similar principles as enzymes. So if you remember, I think way back to the enzymes topic, that enzymes, the lock and key model. So enzymes are specific shaped molecules which combine with a substrate, and the substrate fits into them, into their active site. And the key thing there is specificity, okay? So one enzyme for one substrate, and it's actually exactly the same with antibodies. And actually specificity is a theme that runs all throughout biology. It's one of those key words that comes up all the time in mark schemes. So antibodies are specific to a certain antigen, and you can see here we've got different shaped antigens. These could be different hormones, different drugs, different molecules found on the surface of pathogens, and you can see that they bind like this. Okay, so what are monoclonal antibodies then? So you might be thinking, what's the difference between a normal antibody and a monoclonal antibody? Now, when we think about monoclonal antibodies, we're actually thinking about them in relation to making them in big laboratories on a big scale. And actually now we're not just thinking about the immune system. Obviously this would be made using the immune system with our lymphocytes and all of that. But we're thinking about engineering these monoclonal antibodies for other things. And I'll explain all that in a minute, so don't worry. But as with all things in biology, we can kind of work it out from the name. We can cheat a little bit. So monoclonal. So mono means single and clonal means clone. So monoclonal antibodies are produced from a single clone of cells. And it's going to be the cells that I said that produce antibodies earlier. They were our lymphocytes, our particular type of white blood cell. So it's going to be produced from a single clone of these. And again, I'm going to explain more about that in a minute, so don't worry at the moment. And as I already said about our normal antibodies, that they're all specific to one binding site on one particular protein antigen. Okay, so they're going to be specific to a part of a protein antigen. Just like you can see here, it's not binding to the whole thing, it's just binding to this specific part of the antigen like this. Okay, and I can't hammer home this point enough. They're specific, okay? So our antibodies, our monoclonal antibodies are specific to one part of one antigen, okay? And this means that we can use them to target specific chemicals, and we can even target specific cells in the body, which gives them a huge range of uses. So just before I talk about how they're made, I can see we've got a couple of questions already, so I'll just quickly answer those. So we've got, are antigens and pathogens the same? So that's a really good question, and that's something that students often get confused with actually. Now, antigens are the part of the pathogen that's recognised by the body as foreign. So if we, let me try and draw it out. So if this was our bacterial cell, and it would have some stuff on the surface of it, some receptors, some proteins, and they would be the part that would be detected by the body, by the immune system as foreign. And when we make antibodies to help combat our pathogens, they're going to bind to that antigen, okay? Where are antibodies found? So antibodies would be made by our lymphocytes, which would be floating around in the blood, and they would be released into the blood and they would go to their target antigen. Okay, so right, so that's just a bit about what they are, and this is all going to start to make much more sense when I talk about how they're made. So you probably, if you've looked at this topic before, you've probably seen this diagram crop up everywhere with our mice. So mice are how we make them, and remember we're talking about making these monoclonal antibodies in a laboratory on a big scale. So everything we're doing, we're not just trying to make a few of these. Remember, these can be used for treatments and stuff like that, so we want to make loads. So firstly, what we need to do is we need to get the particular lymphocyte, the particular white blood cell, which is going to produce this antibody. Now, every white blood cell produces a different antibody. And so you've got loads of them in the body. And then when we get a particular antigen, that's going to tell the right white blood cell basically to make loads of itself, and to make loads of itself so that it can make lots of the right antibody to the particular antigen. Okay, so that we're not making loads of the wrong one, really. We just want to make the one that's going to bind to it. So what we do is we inject the mouse with this specific antigen, whatever we're making it for, to stimulate a particular lymphocyte to produce this antibody. And that means that we get lots of these lymphocytes, and we can extract these lymphocytes from the mouse. We extract them from the spleen of the mouse, but you don't need to worry about the details of where we extract it from or whatever. You just need to know we take away these lymphocytes specific to the antigen. And these lymphocytes are good because they're going to make the right antibody that we want. We want to manufacture this antibody, but we're thinking about making this on a large scale. Okay, so we want to ramp things up a bit. We want this lymphocyte to divide loads and loads of times. Now, the problem with the lymphocytes is some cells divide loads and some cells don't. Unfortunately, lymphocytes is one of those cells that once it's specialized, it doesn't really divide very quickly. So this is the clever bit. So what we do is we take a particular type of tumor cell. You don't need to know what type of tumor cell it is. I'll put it on it just in case you read it in your books. I know it's in lots of different books, but I tailored the level of detail that you need to learn in relation to what's on the specification. And on the spec, on the AQA spec, and also others, it just says you need to know we combine it with a particular type of tumour cell. Now, why do we want to combine them with tumour cells? Well, what do we know about cancers and tumours? Well, they arise when we have uncontrolled cell division. And so they're cells that divide really quickly. So we've got these two types of cells here. One of them is making the antibody that we want, and one of them is dividing fast. So why not just bung the two together and make this super cell which is going to do what we want, everything we want to do. And when we combine the two together, we get a cell called a hybridoma cell. Now, a little tip for you to actually understand this: oma means like tumours or cancers. If you think about some of the names of cancers and tumours that you know, you will realise that actually a lot of them end in oma. Okay, so if you forget what this is called, think about hybrid is when we combine things, and oma is because it's got characteristics of a tumour cell. So it does really make sense when you think about it. So we've got this hybridoma cell, which can now not only divide rapidly, it can also make our antibodies. Okay, but we've only got one of them at the moment. Again, as I keep saying, we're talking about scaling this up, doing it on a big scale to make lots of antibodies. So what we want to do is we want to take one of these hybridoma cells that we've made, and we're going to clone them. Okay, I'm going to clone them and produce lots of identical hybridoma cells. Now, because these are all identical, this means that they're all going to produce the same antibody. And this is where it's a monoclonal antibody, because it's from a single clone of a cell. Okay, and then they're all going to produce lots of antibody. We collect that antibody and we purify it. We're going to do lots of things like checking to make sure that there hasn't been any mutations, that it's exactly the right antibody that we want, and it does the right thing that we want. It's going to bind to our antigen. Okay, so that is how we produce monoclonal antibodies. So we do them in mice. We have an antigen. We get our lymphocytes, combine them with a tumour cell, makes a hybridoma cell. We clone that, and then we get all our antibodies. And that is how you produce monoclonal antibodies. Now, obviously I talked a bit around that topic. Now, for your exam, for your GCSE Biology exam, all the points that I've put on the board, this is all you need to know, right? I've looked at the specification, and I'll always check all the different mark schemes and stuff. And this is the most detail that you're going to need to know it in. Okay, and really, it's just about remembering these keywords: hybridoma and lymphocytes. And If you find that you're forgetting that and you want to perhaps learn this topic, there are lots of different ways that you can go about that. I mean, when I did GCSE Biology, I used to just write things that I always forgot, drew a picture of them, stuck it up on my wall or on the toilet door or something like that. So you're always seeing them, and eventually you'll become familiar with the words and it will just be normal to you. Okay, so that's the first part. So we talked about what monoclonal antibodies are. If I just flip back to that page, so antibodies from a single clone, and we've also talked about how we produce them. So if you've got any questions about this so far, drop them in the chat box now. I'm just going to have a read through them. I can see we've got a couple, and I'll answer them before we go on to the second half. So someone's asked what exam board this is. This exam board is AQA GCSE Biology. I had a look, it's also on Edexcel. It should be in most ones. You may not remember doing it because it's sometimes teachers just add it on in the end as like a little side note about monoclonal antibodies. But even if it's not on your specification for GCSE, if you're thinking about doing A-level, you will learn about this, and actually you'll realise that this topic is really relevant at the moment, as I'm going to come on to later. So it is just, to be honest, one of my favourite topics generally. It's really interesting. Someone's also asked if there's a way of remembering all these words and the order they're in, if it asks in an exam. Yeah, like I said, just, well, the usual really, you know, flashcards, stick it up on your wall and try and really understand it, right? There's no point in just memorising things. Try and understand the words. So what I was saying, breaking it down, mono means single, clonal means clone, hybridoma, hybrid means that you're combining them together, oma means tumour cell, and try and remember it like that rather than just memorising things. You'll have a much better understanding generally. What else have we got? Is this for combined science or triple science? So this is in the higher tier. So this is for triple science. Again, it kind of consolidates what you've learned that's just in combined science as well, because it does link to antibodies generally and the structure of them. And I think if you can understand this, you're going to have a much better understanding generally of the immune response and the role of antibodies in the immune response. Okay.
Hi, Olivia. Sorry, before you go on, we've
Yeah.
had some other questions on the Q&A. I think some people who joined a few minutes late were just wondering why there wasn't a maths lesson today. And I just explained at the beginning that there was a bit of a technical issue, but there'll be a maths session. That maths session will be pushed back to Friday this week. Thank you.
Okay, cool. Okay, and finally then, our last question, we've got, why don't lymphocytes divide rapidly? That's a really good question, and generally as cells get specialised, once they've got their functions and they're making loads of the antibodies, sometimes they just lose the ability to divide rapidly, and actually most cells don't divide rapidly. That's why cancer and tumours are a problem, because they gain a mutation that means that they do divide rapidly, and we don't want that. Okay. Does this apply to all animals or just mice? Really good question as well. Generally, we use mice because mice are really easy to use in labs. We use them all the time in research because they're small, they have quite short lifespans, so we can breed them quite quickly. They're easy to genetic engineer if we want to do that, and they're just cheap to keep. So we technically could do it with other animals, but we tend to just do it with mice just because that's what is used all the time, really. Okay. So I think that's all we've got. Do they use rabbits as well? Yeah, they do use rabbits as well. Again, like preference would probably be mice, but in theory we could use rabbits. Are the antibodies from mice only ever combined with tumour cells? If we're going to make these hybridoma cells, we're always going to combine our immune cell with a tumour cell. So yeah. Okay then, so now hopefully you understand the fundamentals of it. We're going to talk about the actual interesting part now. Oh no, before we do that, I forgot about this. Quick question and answer. So have a read of this question, I'll read it out to you, and pick your letter and send it in the chat box, and I'll have a read through, see what everyone's saying. But why are mice injected with antigens? So is it A, so they produce lymphocytes with the specific antibody? Is it B, so they produce lymphocytes with the specific antigen? So they produce tumour cells, C, or so they produce clone cells. Okay, so got lots of letters coming in now. Great to see that you're all answering it. Brilliant. So I can see most people there have put A. A is the right answer. Antibody, antigen can be really confusing. Like get mixed up with them. Sometimes now even I say antigen and then think, oh no, I meant to say antibody. But yeah, try and think antibody, it's this Y shape, it kind of looks like a person. So call it antibody. Okay, brilliant, well done everyone. And second part then, what is the name of the cells produced by fusing a lymphocyte and a myeloma cell? Is it A, lymphoma, B, hybridoma, C, melanocyte, or D, hypodoma? Okay, got quite a range of answers there. Getting lots of B's now. Good, well done, hybridoma. And if you listen to what I was just saying, you would hopefully remember why they're called hybridomas. So that's great. Well done, everyone. Okay, so uses of monoclonal antibodies. So huge range of uses of monoclonal antibodies. Now I'm going to talk through each of these four. We're going to go through some examples, some exam questions. You don't need, like I said, you don't need to actually memorize the uses of them. I'd say if you're going to memorize any, try and memorize pregnancy tests. But what's important, what I want you to take away from this is the kind of key points that you need to include about monoclonal antibodies in your answers in the exam questions about this, because the way it's worded in the specification to say what you need to know is, if I go back to the start, explain how tests and treatments using them work when provided with appropriate information. So, oh, there we go. So give you an exam question. It gives you an example that you might not have heard of before, and you have to read that, pick apart the key points, and show the examiner that you understand what a monoclonal antibody is, and you understand all the key points about it. And okay, right, so we can use them in diagnosis, so pregnancy tests, we can use them in laboratories, we can use them in research, which is really interesting, and also to treat some diseases such as cancer. So let's go through these one by one. Okay then, so monoclonal antibodies in diagnosis. I would say if you've heard of any, you've probably heard of this one. So common example that is seen all the time is pregnancy tests. So monoclonal antibodies can be made to be specific to different things, different antigens. And like I said at the start, antigen isn't always associated with a pathogen. It could just be any molecule really that, and we could use them to HCG, which is the name of a hormone that is made by pregnant women and only in pregnant women. Normal people don't have it. So we manufacture this monoclonal antibody against this hormone, and what we do is we attach it to a pregnancy test strip when we're making these tests. And when you put your urine on the test strip thing, it flows up, and if you've got HCG in there, it's going to bind with this monoclonal antibody. Now, any other molecule that is in the urine isn't going to bind with the monoclonal antibody because it's not the right shape. Okay, so this is an exam question, so a little bit blurry, hopefully you can see it okay. So monoclonal antibodies can be used to determine blood hormone levels. Pregnancy test strips use monoclonal antibodies. The hormone HCG is produced in the placenta in pregnant women. The kidneys excrete HCG. So because they excrete it, that means that we get it in the urine, like I was saying. So we've got 3 different pregnancy test strips here. So hopefully you can read that okay. So we can see we've got a results window, we've got a control window, and here this result control window basically shows if it's a valid result, if the test is working properly, and this window below will show whether it's a positive result. So if you're pregnant and you've got HCG in your urine, hopefully we would get a line here. So which test strip would belong to a woman who is not pregnant? I'll give you a few seconds to say whether you think it would be 1, 2, or 3. Okay, interesting. So we've got lots of 1s, a few 3s. I can see why you think 3. It's too blurry to see. Sorry, what you can see, there's 1 line here in the control window. There's 2 lines here in both the control and the results, and this one, there's no line in the control window, and there is in the results window. Sorry, it's a little bit blurry. That's my fault. Yeah, so the correct answer is number 1, because we've got it in the control window, which shows the test is working, but we haven't got any HCG, so it hasn't bound to our monoclonal antibody, so it hasn't showed up in the results window. Now I can see why a few of you have said 3 now, but that says that there's a positive result in here. There's no result in the valid test window, in the control window. So this one just shows that perhaps the test isn't quite working properly. Okay, so this one would be inconclusive either way. So now we're actually looking at the parts of the pregnancy test strip, and we've been given a few more details about how it works. So if there is no HCG in a woman's urine, she would see a negative test result. Explain how the pregnancy test strip works to show a negative result. So let's have a read of the information that we've given us, and in the back of our minds we're going to be thinking what monoclonal antibodies do, they're specific, all of that stuff. So we've got our sample pad. So we dip the urine here, and what's going to happen is the urine is going to dissolve on it, and it's going to travel upwards, whether that's got HCG in it or not. It's going to reach the reaction zone where we've got our HCG specific antibodies. And these are our monoclonal antibodies that we've manufactured. Now, something as well that's quite important. Sorry, there's something in my throat. Something that's quite important is that we can also attach stuff to monoclonal antibodies, and we'll also talk about this when we talk about their use in treatments. But in this case, we can attach a dye to the monoclonal antibody. So if we do have our HCG, and it's going to bind to this antibody, and it's going to travel up to this results window. Okay, and this is our immobilized HCG specific antibodies. Okay, so it's going to go up to here, and we're going to see a line here, and control window is our non-mobile antibodies complementary to the reaction zone antibodies. So this is always going to show up if basically the reaction zone and if the test is working properly, because if we've got some antibodies there which aren't bound, they're going to bind to them here. So how does this work? Show negative result. So we've got 6 marks here, so there's quite a lot that you can say. So you may need to make sure that you're fully explaining your answers right the way from talking about, so that would underline, so no HCG in the urine, and how does that lead to seeing a negative result in each stage in the test strips. Zooming in a bit, does that make it a bit easier to see? Someone's just asked if I can do that. Okay. Right, so if we're not pregnant, we don't contain the HCG hormone. So when the urine passes through the reaction zone, there is no HCG hormone to bind to the antibody, and therefore any unbound antibodies are still going to move up the stick, and they're going to pass through the results zone. They're not going to bind, so you're not going to have a dye there, and they're going to pass up, and they are going to bind in the control window. Okay, and we're going to see that there. So we would get a blue dye there in our control zone. We've also got, what have we got here? Urine passes up the stick through the results zone and binding, said that. So no dye in the results zone. Mobile antibodies from the reaction zone bind to complementary antibodies in the control zone. So this is where we're really showing the examiner that we understand what monoclonal antibodies are, that they bind to specific things here. And we can see actually these antibodies are binding to other antibodies. So this is where it can be a bit confusing, but antibodies are also proteins. They've got a shape, so they can kind of act as antigens in a sense, and monoclonal antibodies can be made to bind to them as well. So yeah, that's okay, this is an interesting one. So this is linking it to enzymes. So some pregnancy tests rely on the activity of an enzyme attached to the mobile antibodies in the reaction zone rather than a blue dye. So I'm going to draw out here. I always think it's useful when you're reading questions to draw stuff out. I always do it just because I'm not very good at just reading it. I need to see it. I know a lot of students that I teach are the same as well. So some pregnancy tests rely on an enzyme attached to our antibodies in the reaction zone, and that catalyzes a reaction that can produce a change in colour to the test line in the results window. Outline the principles of how both enzymes and antibodies work, both of them. So we need to have a recap of how enzymes work. So enzymes, as I explained earlier, are proteins as well, and they bind to a substrate. That substrate is a particular shape to its active site, and it binds like that, the same way that an antigen binds to an antibody. So if we have a look at our mark scheme, and here's this point which I keep bringing up all the time. Sorry if you're getting bored of me saying specificity, but it's so important. So they both have got this same principle of specificity. So an antibody binds to a particular antigen, and an enzyme binds to a particular substrate. And if you can get in the knowledge there of the active site, then even better. Okay, let's see what else we've got. How would explain how a positive result would be observed in a pregnancy test containing the antibody bound to an enzyme. So we would look back at where is it? I can find it. We'd look back at this, remember our reaction zone, all of that, that's all going to be the same. The only difference is the enzyme. So it passes through this reaction zone. The antibodies with the enzyme is going to bind to HCG and form a complex. It's going to move up the stick still, and it's going to bind to our immobilized antibody, which is stuck at a particular part. And that is where we're going to have like our substrate for the reaction, and it's going to catalyze the reaction there if it has moved up, and that's when we see the change in colour. Okay. Right, so that was first part, first use of them in diagnosis, pregnancy tests. Just have a look at a couple of questions that I've got on that. Can a pregnancy test ever give a false positive result? Well, generally pregnancy tests are really, really accurate. I mean, different companies and people say different things about this, that technically they can't because of this control zone, but I guess that is the occasion where they can. But generally the fact that we have this control zone there does tend to minimize them giving a false positive result. Why does HCG bind with a particular antibody? So really good question. HCG is a hormone, and it's got a particular shape, and we've manufactured that antibody to be able to bind to, we've manufactured the antibody to be a specific shape. Remember the binding site thing, that diagram at the start is going to bind to that particular shaped hormone, okay? So, yeah, if anyone has any more questions on that, drop them in and I'll answer them after. So now a little bit more lab testing, but this time measuring the levels of hormones and chemicals in the blood and detecting pathogens. I'm going to focus on detecting pathogens. So, right, ignore this stuff for now. This is just an example that I put on the screen. I'm going to talk you through that. But let me just give you a bit of background first. So, one that's really interesting is HIV. Now, I know you have to know a bit about HIV at GCSE Biology, and also need to know it at A-level biology too. Now, I know we've talked about it in some other sessions, but if anyone wasn't there, HIV, human immunodeficiency virus. So it's a virus, it's a pathogen, and it's responsible for causing AIDS, which is acquired immunodeficiency syndrome. So HIV basically affects the immune system and all of that. So we need to be, it's really important that we're able to detect HIV in people with a test. And the test is a little bit more complex than just a test strip. We do it in the lab. We do it with something, it's called the ELISA test. Now, GCSE students who aren't going on to do A-level biology, don't worry about memorising ELISA test, anything like that. Just know that they can be used to detect them. If you are going on to do A-level biology, ELISA test is something that you need to know about. So I guess the weird thing about GCSE and A-level is that GCSE, basically, you get a lot of the examples that they use at A-level in the exam questions, and they give you the information, whereas at A-level, you just have to remember a bit more of that information sometimes, and is the case for this topic. So what we can see here is not in the case of HIV, in the case of mumps, which is pathogen. I think you need to know about the vaccine for it as well, measles, mumps, rubella. It's a pathogen that causes a big swelling on the side of your face. Loads of my friends at uni have had it. You do have to laugh about it sometimes. They come out with these huge like things, but not very pleasant. But yeah, so we want to test for mumps if someone's got it because it's really contagious. So this is slightly different. So we've got an antibody. Someone would have a mumps antibody in their blood if they've had mumps. So we can still use monoclonal antibodies, but this time we're using antibodies that are going to be complementary to the antibody. So we're having monoclonal antibodies which are binding to the mumps antibody. If that makes sense. So what we do here, I'm going to zoom in a little bit. So what we can see here is we've got a well in a test dish in our lab, and we've got our mumps antigen. So we've isolated the antigen, which is our bit of the pathogen, and we've stuck it to the well basically. Now what we do is we get a sample of blood. If someone has had mumps, they're going to have antibody in their blood for mumps because it can have antibody in there because they're trying to fight it basically, and especially if they're starting to build up immunity to it. So our antibody is going to, if they've had it, is going to bind to the antigen because antibodies bind to antigens, and then we need to wash it just in case, whether that antibody is there or not, we want to wash it so that so it's just going to get rid of anything that's left, any of this antibody that hasn't bound. And now is where our monoclonal antibody comes in. So we have our monoclonal antibody, which is going to be specific, is going to bind to this mumps antibody. And again, like I said, can have things attached to them. So in this one, we have an enzyme attached to these antibodies, which is only going to bind to the antibody if it's there. If the antibody isn't there, these monoclonal antibodies, they're specific, they're not just going to be able to bind to this antigen. Okay, so then basically we wash it again and we shove in our substrate for the enzyme, which is the stuff that the enzyme is going to react with, and we're going to get a colour change if it's present. So that's how that works. Just another example taken from an exam question. So here are a few things to think about, the kind of thing that could come up in the exam question. Why does this detect only mumps antibodies? Again, specificity. We make our monoclonal antibodies to be a certain shape that's able to bind to the mumps antibodies, which have different shapes to other antibodies, like antibodies for HIV, for example. Why is it important to wash the well at the start of step 4? This is one that students seem to struggle with, and you just have to think about it logically. So at the start of step 4, we wash the well. Well, if we wash the well, if we think about if we hadn't had this, if we hadn't had our antibody in the blood, and it just happened to be there, and we hadn't washed it well, it'd still be floating about. When we stick our enzyme attached to our monoclonal antibody, when we stick that in, that's still going to be there, and it's going to bind with the substrate, even though it's just floating free in the solution because it's not got any antibody to bind to, and that's going to give us a false positive result, and we don't want that. And finally, why is there no colour change if mumps antibodies are not present in the blood? So if there are no antibodies present in the blood, when you do questions like this, you have to go all the way through and logically each step and get your key knowledge in there about monoclonal antibodies. So there would be no colour change because we wouldn't have our mumps antibody because they've not been making it to try and fight against it. So it's not going to bind to the antigen. So therefore our monoclonal antibody isn't going to bind to this antibody. It's a specific shape, it binds to specific things, so it can't bind to the antigen alone. So therefore we're not going to have our enzyme there once we washed it, and we're not going to get a colour change. Hopefully that kind of makes sense anyway. But like I said, don't worry about memorising that, none of that, just get the idea of how you would do it in an exam question. So let's see what we've got next. I've got another example here, but it's really similar. I'm going to come back to that if we have time. I'm going to leave it for now. Okay. This is one that I find particularly interesting because I do biomedical science and obviously involved in a lot of research, medical research and stuff. So in the lab, when we do lots of different experiments, we need to identify where specific molecules are in a cell. So for example, we might be tracking the movement of a molecule, we might be tracking the movement of a particular protein, which we're not quite sure what the function is yet maybe, and we want to see where does it move in the cell when we do certain things to the cell, and huge range of examples we could use there, but we want to identify molecules and we want to locate them. Obviously, if you just look at the cell, you look at under a microscope or anything, you can't see specific molecules, really tiny things. So what we do is we make a monoclonal antibody that is able to bind to whatever molecule we are looking for in the cell. Okay, and we attach that, not with an enzyme this time, but with a fluorescent dye. So when we then put UV light onto it, we can see here, and you've got this lovely picture here, we can see some cells, and we've tracked different things. So it tracks something with this green one, probably the nucleus here, with the blue one and the red around the edge. So we can just see different things, and that's really interesting actually. But to be honest with you, it says that you need to know this, but this doesn't seem to be tested about as much. It's kind of harder for them to do exam questions on using monoclonal antibodies for location. They tend to be on processes like using for diagnosis and for treatments, which we'll talk about in a minute. But there are some questions that are multiple choice, like give two reasons, two things that we can use monoclonal antibodies for, and you could use that here. Okay, and finally then, oh, bit's cut off here, but doesn't matter. Monoclonal antibodies to treat disease. So this one, in my opinion, this is the most interesting and it's most relevant. So cancer is the example that we talk about here most often, but actually you can use monoclonal antibodies to treat a huge range of diseases. Now, let's just have a think about what cancer is. So cancer is the uncontrolled division of cells. And so we might have a tumour or something somewhere, and we want to get rid of these cells. We want to kill them because they can spread, and cancer can spread to other places. So traditional cancer treatment would be like radiotherapy, chemotherapy, but as I'm sure you all know, that has got huge disadvantages in that the side effects are huge because these damage all cells in the body. They, a lot of them target rapidly dividing cells. So like cells in your hair follicles, they rapidly divide. So those would target then. Anyway, I'm going off topic, but monoclonal antibodies for this. So we discovered a much more clever, really, way to treat these diseases scientists realised that cancer cells actually look different to our normal cells, and that's because they've got mutations in them. And so they've got different stuff on the surface of them, so we can identify them. So what we can do is we can take one of the molecules that is only found on the surface of a cancer cell, and we can make a monoclonal antibody specific to that molecule. Now, what we can do is we can, instead of attaching an enzyme, instead of attaching a fluorescent dye, we can attach a treatment, so like a toxic drug, a chemical, a radioactive substance. And this is really clever because it means that this antibody is going to go to our cancer cells and our cancer cells only and bind to them, and then the drug is going to be released, like, and break down or whatever around these cancer cells, and mainly the cancer cells where they are, and a few of them surrounding are affected, but generally not the cells in the body. And you need to know that these types of drugs, these are directed at stopping the cells growing and dividing, because cancer is uncontrolled cell division. So yeah, that's really clever actually, and so we can deliver them specifically, and our major advantage here is that we don't harm other healthy cells in the body, which gives it advantage over our radiotherapy and our chemotherapy. And if we think about why healthy cells are not affected, this is the sort of question that you could get in your exam. Healthy cells aren't affected because, like I was saying, healthy cells have got different molecules on their surface which aren't specific to our monoclonal antibodies. So they're not going to bind, and our drug isn't going to accumulate around our healthy cells. So it's really, really clever. And, you know, cancer is the most common one we see with this, but there are lots of other diseases that we can treat with monoclonal antibodies as well, delivering different things to the cells. And also, I just want to point out with this question that I've put onto the board, we can use monoclonal antibodies just to mimic the function of normal antibodies, really. It's not always about attaching something to them. So this question here is, a virus called RSV causes severe respiratory disease. How would injecting a monoclonal antibody for it help treat a patient suffering the disease? There's only 2 marks here, so you're not going to need to say a huge amount. You're just going to need to use your knowledge of monoclonal antibodies. So any 2 from, the antibody binds to the virus, or it could bind to the antigen on the surface of our virus. The antibody is complementary in shape to that, or specific to it. Okay, there's your knowledge of monoclonal antibodies. That's going to get you 2 marks already. You could go a step further in this if you really understood the immune system and say that our white blood cells are phagocytes. If you remember from the immunity lessons that my tutor have done, the phagocytes could then help kill them, and that would help treating a patient suffering with that disease. So that's just a slightly different example there, but the key thing that you understand is that there are a huge range of applications, and there's no point memorizing them all because there are so many. You just need to be able to understand them, be able to apply your knowledge, think about the key things that you need to know, specificity. It's like the 1000th time that I've said that now, but I'm going to say it again, and all of that stuff really. And that is that. So I think that is pretty much everything that I had to explain to you. So if anyone's got any questions, I can see a few being submitted now. I'm going to go through and have an answer of them. Any questions, any that you need me to explain again, then I will do that. So let's have a look at the questions that we've got.
Having a look, Olivia, I've also seen some come through that I can probably help to answer.
Yeah.
So someone has asked where we might publish these slides and where they can be found if we do. So the session is recorded, and if you've signed up to the My Tutor online school, then you'll be able to be emailed with a link that links out to today's session. So that's how you can access that. I've also seen a question that's asking, Olivia, if HIV is a pathogen.
Oh yeah. Yeah, HIV is HIV a pathogen? Yes, it is a pathogen because it's a virus. Pathogens could be viruses, bacteria, all of that kind of stuff, fungi. So yeah, HIV is a pathogen. What else have we got here? What exam board is this? This is, the exam questions are taken from AQA, but I've had a look at the different examples, and all the stuff that you get asked for, this is pretty much the same between them all. How can we detect cancer cells? That's a good question. So we can also detect cancer cells with monoclonal antibodies. So we can do a similar thing with the antibody specific to the antigen on the surface of a cancer cell, but this time we wouldn't have our radioactive substance. We would have, well, we might have a radioactive substance, but at a lower level that isn't going to kill the cell. Would have a level that can be detected, so that we can then put a screen on that and we can see whereabouts the cancer cells are. What white blood cells produce phagocytes? Phagocytes are a type of white blood cell themselves, and there are different types of white blood cells, and different types of white blood cells can act as phagocytes. There are some that are just, phagocytes are important in the first stage of the immune system, the non-specific immune system, so whatever we get there. Yeah, and they can be sitting around your tissues. Just basically, they sit there waiting, ready for a fight, ready for a pathogen to come in. They're going to detect that and tell the rest of the immune system. Okay, have we got any other questions? Why do bacteria tend inside the body to get alive even when the person is dead? Bacteria inside the body, well, bacteria are cells of their own. So they're cells and they have a metabolism, so they need to respire. So long as they're getting all the stuff that they need to stay alive, so long as they're getting, if they're aerobically respire, as long as they're getting oxygen, as long as they've got glucose and whatever nutrients they need, bacteria can stay alive even if a person's dead or anywhere really, just as long as they've got the stuff that they need to survive. Really interesting question about, are the antibodies for every animal the same? Very good question. I don't want to confuse you too much, but they're not. So if I go back to this, basically there are slight differences between all animals because every animal has evolved slightly differently and gradually accumulates mutations which just slightly change structure of an antibody, not a huge amount, but you would see between different animals, you would see different antibodies, slightly different structures of antibodies. So that's an interesting one. Can you have or produce antibodies for a specific virus if you had a vaccine for it? I think what you mean there is, so if you injected someone with a vaccine with the antigen, and that was to stimulate the immune cells and that was to stimulate the lymphocytes, then you would make antibodies in that person's body. That's different to making monoclonal antibodies where we're doing it in a lab, in a mouse, we're making them on large quantities. But yes, you had a vaccine for a virus, you're always injecting the pathogen, in particular, you're injecting that antigen. Okay. Just seeing if we've got any other questions.
I think we just had one come through, but I'm not sure if it was already answered on the adult stem cells produced in the lining of the intestine.
What type of adult stem cells are produced in the lining of the intestine? So the type of adult stem cell, well, there are different types of stem cells based on how many different types of cells that they can become. So in adult stem cells, we have unipotent stem cells and multipotent stem cells. So depending on where it was in the intestine, if it was just going to make one particular type of the cell, it would be a unipotent stem cell. Someone said, is a dead pathogen put into a vaccine? Yeah, so the key thing with vaccines is that we're always injecting the antigen. The antigen is a specific part of the pathogen which is going to cause the response and cause these specific antibodies to be made. So sometimes we do put the dead pathogen into the vaccine, but sometimes that produces too many side effects. So what they can do is they can isolate the antigen and just put the antigen into the vaccine, or sometimes they can just weaken the pathogen, and it depends really, but yeah, we can put the dead pathogen into a vaccine. Someone said, I remember during enzymes, the shark eating a slice of pizza. Is this a good way to remember during enzymes? Yeah, it is a good way to remember during enzymes, because I guess the shark's mouth would be the active site and your pizza would be eating it, and you just got to remember if it was eating something else, wasn't a pizza, that would be a different structure, and therefore we would need a different enzyme because of specificity. And the shark's mouth would have a different shape because the active site would be a different shape. Are T cells phagocytes? No, so T cells and B cells, it's not something that you need to know at GCSE but too much, but you do need to know it in more detail at A level. So lymphocytes are B cells and T cells. They're T lymphocytes and B lymphocytes. And they're different. They're part of the specific immune response to a specific antigen. So they're not phagocytes. Yeah, so T cells are involved in killing the pathogen directly. They're part of cell-mediated immunity. After getting divided by mitosis, do stem cells become specialized? Yes, they do. So stem cells can do two things. I know it's not relevant to this, but stem cells can firstly divide and to make more of themselves, and secondly, become specialized. Oma is tumour. Yes, oma does mean tumour, and that's why we had our hybridoma and our myeloma. Does GCSE differ with IGCSE biology? Yeah, there will be slight differences. You would have to have a look at your specification and see exactly what it is that you need to know on this topic for IGCSE biology. Most of the principles at GCSE are pretty much similar. It's just little differences in wording and, you know, key little bits that are going to get you the marks on the mark scheme. So just make sure if you do IGCSE biology that you go and have a look at your specification and just compare it really with what you need to know. It's probably quite similar. I think that's all our questions for now, unless anyone has any more.
I agree, and I think that's probably a good place to leave it here, as we've only got a minute or so left. But thank you so much to everyone for joining us for this one of our first biology GCSE higher sessions. So look out for the timetable next week to know what's coming up in terms of biology, and also for the rest of the sessions for this week. Thanks, Olivia, and thank you, everybody. Bye.
Thanks, everyone. Bye. Hope that helped.